Hello, everyone. My name is Khushi Patel, and the title of my research paper was Targeting LDLR for Precision Treatment of Atherosclerosis. And my mentor was Kadeen Edwards. So, my research question was how can changes in LDLR levels at different stages of atherosclerosis, including differences among European populations, be used to inform new treatment strategies? So on the next slide, I have a little bit of background regarding my research question so we can make it easier to understand. So I first mentioned atherosclerosis and LDLR. So LDLR is a gene and protein that is responsible for removing LDL cholesterol, which is also known as bad cholesterol from the bloodstream. Now, when LDLR function is reduced or there's mutations, LDL accumulates and contributes to plaque formation. Now, atherosclerosis occurs when fatty plaques build up, narrowing arteries and restricting blood flow. Now, this restricted blood flow can lead to heart attack or sometimes even strokes. Now, in my research question, I also included differences among European populations, but these differences can also be based on age, sex, lifestyle factors, and also where you live. So these differences explain why treatments such as statins, antiplatelets, PCSK9 inhibitors don't work for everyone equally. So that's why you need to have something that can work for people if all these other treatments don't work. That's why I developed the research on LDLR. So then I also narrowed my research question down a little bit to European populations because before it was including variations among populations, which is really broad, as I mentioned with these ones. And lastly, I included how LDLR could be used to treat atherosclerosis on the molecular level. Then, this research paper is done on a study based on systematic review of existing research, and some sources included peer-reviewed studies, review articles, and clinical data. So, my focus areas for this research was LDLR functions at different disease stages, genetic and population differences, and responses to LDLR targeting therapies. Additionally, it was harder to conduct the research literally in a lab with different kinds of people because it's a genetic research. So, you would have to do things on the molecular level. So, some of my key findings were these four things that I discovered from reviewing a lot of articles. So, I discovered that timing matters. So, LDLR boosting therapies work best early in atherosclerosis, and in later stages, plaque structure and inflammation reduce their effectiveness. Next, you need to personalize the treatment. So, genetic differences in LDLR affect how patients respond to treatment. Like if someone has a mutation with LDLR, increasing its flow wouldn't help the mutation. So, that's why I need to personalize the treatments. And then some individuals benefit significantly while others see limited effects. So, that's why the third result was a combination approach. So, best outcomes occur when LDLR targeted therapies are combined with statins, PCSK9 inhibitors, and this can reduce things such as inflammation and improve metabolic health. Next, you need to consider different populations. Like over here on the previous slide, I mentioned there's a lot of variations, so you need to take into account all of these variations when you develop these treatments. Additionally, some patients may need to start the LDLR treatment earlier or need to use more of it to get rid of their atherosclerosis than other people, like the European populations. They have high LDL, so they may need a more aggressive treatment of LDLR. Next, some of the difficulties that I encountered in doing this research was understanding the literature. Sometimes the way things are written or the way things are worded in some of the reviews or some of the articles can be a little difficult to understand. Next, in my paper, I had a little bit of trouble with grammar, commas, apostrophes, things like that. I had a little bit of difficulty with that. And then searching for research gaps, so things that haven't already been researched, had a little bit of hard time doing that. And then, even though you get a lot of time to complete your research paper and all of the things in between, there was still like a time pressure, like I have to get my research paper done in 1 month and things like that. And sometimes finding reliable sources and references that were in the timeline that I wanted to find reviews that were done in the last 10 years to be more specific and, you know, things like that. So it was a little hard to find sources in between the timeline and things that are credible. Lastly, I want to emphasize that research is collaborative and it cannot be done in isolation. So, I'd like to thank my mentor, Katie Edwards, for the guidance and feedback, and then also my program manager, Kabir Hirandani, for all the structure and support of the program, and also my writing coaches. I am very thankful for that because, as I said, I struggled with grammar. So, thank you for helping refine my paper and provide scientific clarity. And that is all. Thank you so much.